Hello, everyone. My name is Jeff Rothstein. I'm a professor of neurology and neuroscience at Johns Hopkins School of Medicine. I run the ALS research program in the clinic. ALS is largely a sporadic disease affecting middle-aged individuals, and it's a uniformly progressive and fatal disease. For years, we as a community have tried to develop new drugs for this disease. And to be honest, we do not have strongly effective drugs. In part, this reflects the fact that we guess on what could be wrong in ALS by looking at perhaps blood markers in mouse models, but we have lacked until recently the ability to truly sample the nervous system of our patients the way we've very successfully done in cancer. And many of you know in cancer, we're able to biopsy the cancer, examine its molecular and biochemical properties, and in so doing, come up with pathways that can be even individualized to patients. This has been well established in melanoma, prostate cancer, and breast cancer. That biology has now changed so that we can do almost the same thing in ALS through the use of a new approach, the generation of induced pluripotent cells. Through sampling of patients' blood, we can reprogram those blood cells into stem cells, which are then eventually programmed into the patient's spinal neurons, the very cells that degenerate in ALS. In so doing, we have then a biopsy of the patient's nervous system, and then we can carry out the very similar kinds of approaches that have been done successfully in cancer, including whole genome sequencing, proteome, epigenome, RNA analysis. And to carry this out in patients, we devised the program Answer ALS, in which we've enrolled now 1,000 patients, initially at Hopkins, but around the country, followed them for well over a year, collecting deep clinical data. In fact, we developed even an app for the iPhone to collect even more data, as this is a progressive disease and often leads to patients failing to come to clinic because of their progressive disability. Their blood was converted into these iPS cells. The cells are then converted into neurons, and the biological analysis I've mentioned is carried out. In total, this amounts to more than 6 billion data points per patient. It's a lot of data, and the cells are a living example of the patients, and together, all of the data and the cells are now freely available through our Answer ALS web portal, which is available to all researchers, both academic and commercial. And the point of the program is really as a foundation program, so that this data and the cells can be used to discover pathways in ALS, and because we have living cells, to use them as a platform to discover new drugs. Because Answer ALS shares overlap with pathways that are seen in Alzheimer's and other dementias, this data will be equally useful for those researchers, both commercial and academic, that are interested in trying to uncover new therapies for not just ALS, but other neurodegenerative diseases. Thank you.